Good morning, students. Though I was taking the online class but could not manage, there was a voice problem also. It was cracking, as you said. So now I am making the video. Please try to understand the circulatory system, and that circulatory system's next part we will see about the blood vessels. So blood vessels are the branch tube extending from the heart, that is artery, veins and capillaries. So once again I say here, the blood vessels are the branch tube which extend from the heart, and they are artery, vein and capillaries. So first we will see about the difference between the arteries and veins. Arteries and vein. Here the artery, you will be asked the question like this: write two structural difference between artery and vein. So sometimes you will be asked to draw this diagram. So how to draw this diagram? First, whenever you draw, you have to make a narrow lumen. Narrow lumen. Here in vein you have to make wider lumen. Narrow lumen, wider lumen. Here you can see the endothelium. Endothelium, same in the artery also. Then there is a smooth muscle. Smooth muscle. Here I have written smooth muscle, that is middle layer, and the external layer, that is connective tissue. Connective tissue. External layer, connective tissue. And next is, so you will be asked to write the difference between arteries and veins. So structural difference, especially when they ask structural difference, you have to say artery is having narrow lumen and the vein is having wider lumen. One difference. Second difference, artery is having the thick muscular wall. You can see from here to here the diameter, and this diameter you can see. It is having the thick muscular wall, and vein is having the thin muscular wall. So these are the two structural difference you can write when you are asked this question. And next, blood flows in artery in spurt because of ventricular systole, and here the blood flows uniformly. Why? It is having the white lumen, and here in the lumen there is in artery you can see the valve. This is the valve which facing towards the heart. So blood flows towards the heart from the vein. Here there is a valve which prevents the backflow of blood. Preventing the backflow of blood. So this is the vein, having the pocket-like valve here which prevent the backflow of blood. So blood flows here uniformly. So these are the differences between arteries and veins. And next we will see about the capillary. Capillary. This is the fine branch of the blood vessel, providing the blood to the whole part of the body and to each cell, we can say, of the body, to each cell. And this capillary is having the many function. We'll see the function also, like suppose this is a capillary. So it is running in the body, providing deliver the oxygen, outward diffusion of oxygen to tissues, cells, etc. Outward diffusion of oxygen. When oxygen is used in breaking down the food to release energy, carbon dioxide and water, that carbon dioxide is diffusing inward. Carbon blood. So this vein carries again back to the heart, and there from heart it is pumped to the lungs for oxygenation. Carbon dioxide we breathe out, and again it gets the oxygenated blood. So these are the function of the capillary, that diffusing out of the blood vessel, that is oxygen for the tissues and cells, and carbon dioxide in turn diffuse in, and also many usable substance like amino acid, glucose, urea diffuse in and out. The waste material will diffuse in, and useful things will diffuse out of the capillaries to the cells and tissues. And then these capillaries reunite. This is the branch to the whole body cells, supplying the nutrients, oxygen, nutrients in that contain the oxygen. Even the white blood cells also diffuse out when it is necessary to kill the germs, to destroy the germs. White blood cells also leak out or diffuse out from the capillary. So, after all these blood vessels reunite to form the venule, which again assume the three layers what we are having in arteries and veins. Capillary is made up of single layer of epithelial cell. Sometimes you may be asked to name the blood vessels which is made up of single layer of squamous epithelial cell. So squamous epithelial cell, it is of single layer, but when this capillary again reunite to form the venule, I will show in the diagram just. So I wanted to say here the capillaries. Can you see the capillaries here? How it is supplying the blood to the cells and tissues everywhere. Then this blood vessels, capillaries, veins, now they reunite to form the venule. This is the venule which again join to the posterior vena cava in the lower part of the body, and upper part of the body, that is the superior vena cava. This is superior vena cava and this is posterior vena cava. So these veins are rejoining to form the venule which joins in the again to posterior vena cava. From this is from lower part of the body and this is from upper part of the body, that is superior vena cava, and again that enter into the heart. Right auricle, from right auricle it goes to right ventricle, from right ventricle it is pumped to the respective lungs, from the respective lungs again pulmonary vein enter to the left auricle, and from left auricle to left ventricle, and again it pumps the blood to the whole part of the body. So this happens. Now blood circulation, that is systemic and pulmonary blood circulation. Here we will see about the pulmonary blood circulation, that is from the heart to lungs. Means blood is from right ventricle pumped to the respective lungs, and from respective lungs after oxygenated comes back to the heart. This is the pulmonary blood circulation. And systemic blood circulation, I have already mentioned to you that the blood from left ventricle pumped to the aorta divides and redivides, forming the arterioles and finally the capillaries, and then it is providing the whole part of the body, and from whole part of the body the vein recollect the deoxygenated blood and bringing back to the right auricle. From right auricle it goes to the right ventricle, from right ventricle it pumps to the respective lungs, again it continues. So this is the systemic blood circulation. And next we will see about the hepatic portal vein. Here you can see from stomach and intestine, blood vessels first enter to the liver. So before here you can see the blood capillaries, fine blood capillaries, which comes out from stomach and intestine, reunite to form the hepatic portal vein. You can see in your book also, this is the hepatic portal vein, and hepatic portal vein enter to the liver. In the liver again it redivide into fine capillaries, and when it comes out from the liver it reunite to form the hepatic vein. Hepatic portal vein when comes from the intestine and stomach and reunite forming the hepatic portal vein, and then enter to liver, again breaks out, and then it is before coming out reunite to form the hepatic vein, and that joins the posterior vena cava. It joins the posterior vena cava. So one question here, why these blood vessels first enter to the liver? So in liver what happens, if there is excess glucose in the blood, that will be converted here into glycogen, and if there is some toxic substance, that will be detoxified by the liver. That's why first this blood comes along with nutrients to the liver, then enter to the posterior vena cava and goes to the heart, right auricle, from right auricle right ventricle, and forms to the respective lungs for oxygenation. And then so sometimes you will be asked to define what is hepatic portal vein. So hepatic portal vein is the vein which arise in the form of capillaries and ends in capillaries. That is hepatic portal vein. And next we will see about the pulse. And the pulse, you will get to define pulse, that is pulse is the alternate expansion and elastic recoil of the walls of artery during ventricular systole. So it is the alternative pulse. You can just press here in the radial artery which facing the thumb. Here you can feel coming up and down like that. So that is the pulse. Pulse counting is indirectly counting the heartbeat. So how to define the pulse? Pulse is the alternate expansion and elastic recoil of the walls of artery during the ventricular systole. Up to that you have to write. Alternate expansion of the walls of artery and elastic recoil during the ventricular systole. So please learn the definition. And next we will see about the blood pressure. It's just an information. The blood pressure, that is, you know that blood pressure, here I will write systolic is about 100 to 140 mm, and diastolic is 80 to 90. This is normal blood pressure. If above that and above this causes the hyper blood pressure in the body, hypertension. So please remember this. Accurate average blood pressure is about 120 systolic to 80 diastolic. This is average, but the normal comes under this 100 to 140 mm systolic and 80 to 90 systolic, that is diastolic. And next we will see about the tissue fluid. Tissue fluid or intercellular fluid. Tissue fluid or intercellular fluid. So through the blood capillaries, the tissue fluid, here just suppose this is the blood capillary, here cells and tissues present. So from the blood capillary, plasma and leukocyte leak out, and that baths the cells or tissues. It baths the cells or tissues. So this plasma or leukocyte which coming out from the blood capillary, that is known as tissue fluid. That here it is known as tissue fluid or extra intercellular fluid. So this tissue fluid, some of this tissue fluid enter back to the blood capillary. Some enters into new channel. Some enter into new channel, that is lymph vessel. Lymph vessel. In the here it is not shown, but in your book it is very clearly shown. I said leukocyte plasma that leak out from the blood capillaries and baths the tissue or cells. And in turn, carbon dioxide from here diffuse into the blood capillary. Some of the tissue fluid is used by these cells and tissues. Some is reabsorbed by blood vessel, and some enter into new channel, and this here it is called lymph. Here it is known as now lymph. So this lymph flows in lymph vessel. There is another channel. Here you can see, I will show once in the book. Here you can see the another channel is there present here, that is lymph vessels. So this lymph fluid flows in this lymph vessel by the contraction of surrounding muscles. When surrounding muscles contracts, this fluid flows in this vessel, and on the way there are some nodes, lymph node present here. So this lymph fluid is drained into the lymph node, and from lymph node, here some places there is lymph node. From lymph node arise the new channel. Lymph node arise the new channel, and that ultimately joins the posterior and superior vena cava, which again comes back into the circulation. So some lymph glands are there, like here tonsil is there, that is example of lymph gland. And in the body many places you have seen that lymph gland is present, lymph node is present, that from there new channel arise and join to the posterior vena cava. So this again it is put back into the whole circulation. And next we will see about the spleen. Spleen is a large lymphatic organ located just behind the stomach, and above the left kidney. Size is about this size, clenched fist. And next we will see about the function of the function of the spleen. So location is behind the stomach and just above the left kidney, spleen. Function, it acts like reservoir of the RBCs. It produces RBCs in embryonic stage. So you learn at least one or two function of the spleen. And next we will see about the examination of blood cells. For this what we have to do, we have to wash the finger and prick the sterilized needle, and first drop you must wipe. You have to take two slide, glass slide. You have seen the glass slide, I have already shown in the lab, and squeeze from there and take a drop of blood in the slide, glass slide, drop of blood. You have to take another slide and swipe this. So the blood will spread. Blood will spread in the slide. You need to leave it to dry. Then add the Leishman stain. That is a chemical which will add the color there, and let it dry. Then wash under the tap water, and then after drying the slide we have to see under the microscope. So we will see the different types of blood cells, red blood cells, white blood cells, platelets, types of white blood cells, etc. So this way we can examine the white blood cells. Sorry, all the types of blood cells. Now, here I complete my lesson, and the chapter is completed, and this is coming in the first term also. So syllabus is completed for the first term. Dear student, please go through it, do the exercise. Any doubt or any query, you can call me in any time, and if possible online class, we will have the direct contact with you. Today, since it was not possible, I did stop, and now I completed the lesson. Thank you.